Thank you, Eugene, and thank you for the organizer for the invitation to speak today. It's an honor to be here. And I would like also to thank for the live streaming, because my family is also watching before they go to bed. And it's a hard job to come after the internet, but I will try my best and talk to you about immunology. So since the discovery of HIV, immunologists have been working really hard to try to understand how the immune system battles against HIV. And over the last 35 years, they made a lot of progress in the knowledge of this fight between the immune system and how we can improve it for treatment and prevention. And so today, in this ABC of HIV immunology, I will try to show you the main discoveries of HIV pathogenesis, which I think we have comprehended quite a bit. And then I will also talk about how we can induce immune responses for HIV remission strategy and what type of immune responses we need to induce. And finally, I will talk about immune responses for HIV prevention. So the first battle between the immune system and the HIV is at the portal of entry in the genital mucosa. And it has been shown that the immune system status is very important for this battle, as the genital mucosal inflammation status increases the risk of HIV infection. Once the virus enters the mucosa, the infected founder population is infected and rapidly recruits other CD4 T cells that are fueling the infection. And then it becomes systemic when they reach the bloodstream. And the studies that have looked at the immune system in the mucosa have been conducted mainly in monkeys, and they have shown that the immune responses, including the HIV-specific CD8 T cells that are here, were actually too little and too late to really combat this first infection and limit the spread of the infection. They have also shown that greater early CD8 T cell responses against the virus in the mucosa were associated with a greater reduction of viral load. So one of the main features of HIV pathogenesis is actually global immune activation. This global immune activation is first driven by HIV replication, but I will show you that it's sustained in a vicious circle. So HIV replication drives immune activation, which then also HIV replication depletes CD4 T cells that are in the gut and that are containing most of the T cells in the body. This gut mucosal damage drives mucosal translocation, and that translocation induces more immune activation, which feeds back to more HIV replication. And also, importantly, immune activation leads to immune deficiency, which leads the opportunity of having opportunistic infection and also fails to control HIV replication. And then it goes on as a vicious circle. And this immune activation has been very important in HIV disease. It was shown that immune activation markers are better predictor for disease progression than the magnitude of the viremia of HIV. So there were two types of immune controllers that were defined and described, and they were also studied. They are elite controllers and post-treatment controllers. The elite controller have an estimated frequency of less than 1% of the HIV-infected people, and their primary infection is rather asymptomatic with low viral load and high CD4 counts. And they have been described to carry HLA alleles that, for the vast majority of them, are protective alleles like HLA-B57, B27, or B58. And because of these HLA alleles and other studies that we have done, these elite controllers are suspected to have an immune control mediated by CD8 T cells that are recognizing HIV epitopes through these AMHC class 1 alleles. Another type of immune controllers that were more recently studied are called the post-treatment controllers. And depending on the studies, I should perhaps have put a 0 here, but there are various frequencies depending on the studies. But these are people who started treatment in primary infection and were remaining on treatment at least for 1 year. In primary infection, they were very different from the elite controllers. They have symptomatic acute with high viral load and low CD4 counts. And for HLA alleles, in contrast to the elite controllers, they have HLA alleles that are associated with high viral load. But recent studies that were presented in Amsterdam at the AIDS 2018 conference showed that actually those HLA alleles that were associated with higher disease progression were actually associated with a better natural killer cell function, suggesting that the immune control in those post-treatment controllers would be mediated by natural killer cells. But for the vast majority of HIV-infected people who don't have these genetic characteristics, there is no control of the virus, and we don't exactly know which type of immune responses we need to induce for prevention and treatment. But we know a little bit of some of the characteristics that I will go to describe. So for prevention, we need immune responses that can block one virus at the site of entry, because we know that the infection is seeded by a single founder virus. In contrast, for treatment, the virus is hidden in many different places in the body, and so the immune responses have to block a swarm of viruses at multiple sites of the body when ART is interrupted. And so therefore, for treatment, we need to have an immune response that is very potent and in the whole body to try to fight these viruses that are recurring. In contrast, another characteristic that we know we need for immune responses for prevention is we want these responses to be persistent over time, because there will be a delay between the vaccination or the intervention and the time where the person encounters the virus. So we need those immune responses to stay and persist over time. And finally, we need immune responses that are targeting a various number of different viruses. So for treatment, we know that the virus evolve in the body, and so we need immune responses to be able to target those different HIV variants in a single individuals. And for prevention, we need even a broader immune response to combat the multiple HIV clades that are represented here in these phylogenetic trees, because we don't know what type of virus the individual will encounter. And so a couple of years ago, one hypothesis that was proposed is that by treating early, we could potentially prevent immune damage induced by HIV infection and by global immune activation. And so the global immune activation occurs relatively early in infection at the end of the acute infection, but whether treating earlier in acute infection would prevent immune responses and it would then lead to immune control after treatment interruption was not known. And this was very difficult to test, because that would require to detect, recruit, and then treat people in very early stages of acute infection. And this was actually done here in Bangkok at the Thai Red Cross in the RV254 Thai cohort of adults that were recruited in the earlier stages, and you've heard earlier this cohort presented by Dr. Jintanat. So we showed that in this acute infection cohort of people that have been treated in the earliest phases of acute infection, the immune functions were actually preserved, and they were keeping a good immune function that were not still damaged by the infection and the global immune activation. However, these functional immune responses were not sufficient to prevent viral rebound, as it was shown here by the different people that were enrolled in treatment interruption studies that show a viral rebound after treatment interruption. So these data showed us that if people don't have the genetic characteristic to have their immune system control the virus, it will be very difficult to gain HIV remission through the immune system control, and we need other immune intervention to drive this control. And so let me talk to you a little bit about how we need those immune responses in HIV remission strategies. So the first strategy is to induce immune control when we stop treatment. The second strategy is because the HIV-infected cells during ART are not expressing HIV antigens, they are not recognized by the immune system, we need to reactivate the virus with latency reversing agents for them to re-express HIV antigen and be recognized. And this is called the shock and kill strategy. Then we need to boost the immune system to be able to eliminate better those reactivated infected cells. And this is called the shock and kill strategy. There is an additional strategy that is starting to be explored, and which is called the block and lock, and which consists in blocking the proviral promoters and then lock the expression of that virus. And this strategy does not involve immune responses, so I'm not going to talk about it. So there are multiple ways to target the HIV reservoir for by immune responses. The first way is to induce vaccines to try to boost those immune responses and to have them recognize HIV-infected cells to eliminate them. Another tool is broad denaturizing antibodies. These are antibodies that can recognize different variants of HIV, and they can recognize HIV-infected cells and then can target immune cells to eliminate them, especially by NK cells. There are additional strategies like the dual affinity retargeting therapy or even gene expression or genetically engineered T cells, where CD8 T cells are transformed to express a receptor that can redirect them to recognize HIV-infected cells. But these two strategies are really less far along, and I'm not going to talk about them today. I'm going to focus about vaccines and broadly neutralizing antibodies. So in the last few years, a lot of bNAbs, or broadly neutralizing antibodies, have been generated, and you can see the list here that are recognizing different binding neutralizing epitopes on the HIV trimer of the virus. These antibodies have various breadth and potency depending on each antibody, and there are at least 8 that are already in human studies currently, and this allows for the combination of 2 or 3 broadly neutralizing antibodies. There are also modifications to make them persist longer in vivo when we infuse them. And the passive infusion of bNAbs for treatment is a high bar, because I said they need to target a lot of viruses that are going to reoccur in different places of the body. In monkeys, the remission has been more successful, but in human, the infusion of bNAbs have shown minimal delay in viral rebounds and minimal reduction of HIV reservoir. And as you can see in these two graphs of a study that was published last year, the people that were carrying resistant virus to these antibodies that were tested here, 3BNC117 and 101074, there was no impact on the delay to viral load rebound. Only in the individuals that had a reservoir that was sensitive for neutralization by these antibodies showed a delay to viral rebounds. And these data show that the response depends on the antibody susceptibility, but also that 2 antibodies are better than 1. As you can see here, this is the combination of the 2 compared to the red, where it's only 1 antibody. But perhaps 3 antibodies will be better than 2. For the therapeutic vaccine, the bar is really high too. There are over 100 vaccines that have been already tested, and that the vast majority of them had showed very little effect. These are 2 studies that were either published or presented last year at the HIV/AIDS conference in Amsterdam. Here it's a plasmid DNA vaccine that showed that there was absolutely no difference between the placebo and the treatment arm receiving the vaccine, but also showing the importance of having a placebo in those clinical trials. This is the RIVER trial that was testing a shock and kill strategy with the vorinostat as a latency reversing agent and the chimp ADS3-MVA as the vaccine. And this study showed no difference between the placebo and the treated arm for the frequency of replication competent virus. So we know that there are other things that we need to restore in order to boost those immune responses. And the first one is try to restore immune function. As we know that chronic infection induces immune checkpoint proteins that are associated with impaired function. This is represented here by the expression of PD-1 on the exhausted T cells, that when it binds to the ligand PD-L1 blocks the TCR signaling, and this cell become exhausted. But the blockade of these immune checkpoint blockers rejuvenates the T cells. In this example, if we block PD-L1 here, the TCR signal is allowing the reinvigorated T cell to proliferate, survive, and have a better cytotoxicity. So another thing that we can do is we can try to target sanctuary sites of a reservoir for HIV. T follicular helper cells are located in the germinal center in the lymph nodes, and they are providing the help for B cell to mature. But we also know that the T follicular helper cells are the major reservoir of HIV in the lymph nodes. But these cells are not accessible for other immune cells, like the CD8 T cells that are located in the T cell zone. And so they reside in a as we call a sanctuary site, and the reservoir is hiding there. So immune strategies to disrupt those germinal centers or help immune cells to migrate into those sanctuary sites would have a better efficacy for immune responses. And we know that the immune response in the lymph node is very important, as lymph node T cell responses have been shown to predict the efficacy of live-attenuated SIV vaccine in monkeys. And lastly, the use of adjuvant to boost immune responses can also be useful to have a better response. So TLR receptors, Toll-like receptors, or TLRs, are receptors that are expressed by innate immune cells to sense microbes, and they're used as adjuvant to increase immune responses to vaccination. And in these two examples here, the TLR7 agonist provided an adjuvant effect in the monkey model to lead to the remission in some animals. And here it was used in the combination with the vaccine at 26 MVA, plus the TLR7. You can see in the red line here that the monkeys had a little bit of delay, but they had also a significantly lower viral load set point after treatment interruption. And in this study here, the broadly neutralizing antibody PGT121 in combination with TLR7 agonist here in the blue line showed a better time to viral load rebound compared to the other groups of animals. So all these strategies are currently being tested, and the field is very dynamic and generates a lot of data, but it will be very hard to have an HIV remission. So we need all these tools to do that. For immune responses for HIV prevention, we can use the same tools. We can use vaccines to try to induce immune responses to battle the HIV at the site of infection. And we can also use broadly neutralizing antibodies that can be good tools to block the HIV from entering and seeding the infection. So the bNAbs for prevention are a lower bar than for treatment intervention because, as I said before, they need to block only a single HIV entering at a single site. So the AMP trial is a phase 2b trial testing one bNAb, VRC01, that is targeting the CD4 binding site. And this is conducted in several places in the Americas and in Africa. And this consists in injecting VRC01 every 8 weeks for 22 months. And so the results of this trial will be available at the end of 2021. For vaccines, we had the STEP trial that in 2005 was initiated that was using an adenovirus 5 virus vector. And this trial was stopped in September 2007 after the interim analysis showed no protection and even an increased risk in the vaccinees with high adenovirus 5 antibody titers. Further analysis of the responses showed that the vaccine induced low HIV-specific CD8 T-cell responses and limited breadth. And so from this trial, we could really take the lesson and know that we have to combine vaccines that induce T-cell responses, but also B-cell responses and antibodies. And we have to have vaccines that induce better, higher magnitude and better breadth of the T-cell responses. So the adeno26 viral vector is one example of these lessons. This is another virus vector with another strain of adenovirus, and this was tested in the APPROACH trial, where it was in combination here with the GP140 in the vaccine regimen. And you can see in the human trial, as shown in the blue line, the vaccine combining at26 mosaic HIV and GP140 induced very good responses. And these responses were persistent after 1 year after the last boost of vaccination. The same regimen was used in monkeys, as you can see in the orange line here, that showed really similar responses to the vaccine. And these monkeys, after the last boost, were actually challenged with SIV, and they showed 67% protection, giving good hope that this vaccine could also induce some protection in humans. You can see in the challenge vaccine in the monkeys here, the turquoise line is the at26 GP140 vaccine regimen and was better protected than the other groups in this vaccine. So the same vaccine is actually tested now in a phase 2b trial, that is the HVTN705 in South Africa, and the results should be available by 2022. Another type of vaccine that is not actually looking at inducing memory T cells, but more effector cells at the site of infection is the cytomegalovirus or CMV-based vaccine. This was tested in the monkeys with the RESIS-CMV vaccine. And this vaccine is given before infection, and it doesn't block infection and entry of the virus, but it induces an immune-mediated control of infection. As you can see here, some monkeys, 55% of them become controllers, and actually the virus cannot be recovered in those monkeys that are controlling. So the vaccine induced 55% efficacy, and it has been shown that the control in these monkeys require high frequencies of non-conventional MHC class E restricted CD8 T cells. We don't know yet if the human version of that vaccine will induce the same responses, and a human trial is about to start in 2020 to look at that. But I would like to end with the only vaccine so far that has shown and induced a potent immunity, which is the RE144, as called as the Thai trial. And as you can see here, after 3.5 years, the vaccine showed 31% efficacy. So in this vaccine, after 1 year, it induced 60% of protection, but the protection decreased quickly to 31% after 3.5 years, showing that the immune responses were not persisting enough. But having a vaccine that really gives people that were protected versus people that were not, were able to then look at the correlates of immune protection. And these were published in 2012. Based on these immune correlates of protection, a different vaccine regimen was designed for increasing the efficacy and prolonged the persistence of the immune responses. And that consisted in giving an extra boost using a new adjuvant, MF59, and this vaccine was adapted for the subtype C virus. This new vaccine regimen was tested in the HVTN100 study that showed that the vaccine regimen was safe and immunogenic, and this was published just last year. And now this vaccine is tested in a big phase 2b, phase 3 trial, HVTN702 in South Africa, and the results should be available around 2021. So there are also a lot of phase 1 trials for development of other vaccines. I didn't touch on those ones because these were the vaccines that are more far along. But from all these vaccines, we can really learn a lot about the immune protection, the immune responses that need to be induced. And hopefully in the next 5 years, we could have the last letters of the alphabet for the ABC in HIV immunology completed. And hopefully by 5 years from now, we can close the book and have a complete story of what we need to induce as immune responses for HIV in prevention and treatment. And with that, I would like to thank MHRP leadership, especially Dr. Jintanat, who is of tremendous help for everything I do, the collaborators, and thank you for your attention.
Thank you very much, Dr. Lydie. I think I'm not the only one in the audience that's very grateful for you bringing down complex concepts to very understandable presentation. Any questions in the audience? Please feel free.
Lydie, this is Mark. I have one question. In terms of the long-term non-progressors, has anybody looked at the ethnic distribution? Is there a propensity for any sort of ethnic grouping of long-term non-progressors versus elite controllers? Are there differences? Race differences, racial differences?
I don't think there is really any data on these type of things. And I think it would be very good to have a really global analysis of those people, depending on the clades and depending on the age and all these other. But this has not, a meta-analysis, if you will, of these elite controllers has not been done. That's a very good point. Thank you, Mark.
Lydie, thank you. I just wanted to add a little bit because we had a chance to talk to Asir Sayzirian.
Sayzirian, yes.
Yes. Okay. So the Visconti cohort that showed 15% post-treatment controllers, and he found an NK cell-related allele that was associated with the control. And he hasn't published this, but he told me that they did some screening in Asians, and it's very low.
Yeah,
the post-treatment controller allele is very low. So I think that one thing that we want to work with him also is to see in the Thai cohort how much we see of that.
Absolutely. But, and I should add that these post-treatment controller cohorts were found post-hoc, really going back to samples. And now they are trying to conduct a new clinical trial where they would enroll those people that have the associated alleles and see whether they can have, in a prospective cohort, the same kind of percentage of immune controllers. So that's not known as now.
One question, forgive my ignorance, but the HVTN trial you showed today optimized the vaccine for subtype C. Assuming if it's effective, you have to optimize it again for other subtypes. That CMV vaccine, is that similar? Does it need to be optimized per subtype, or would that be more broadly effective?
Well, the vaccine really depends on the antigen you put inside. Yeah. So there is, for example, the Ad26 MVA has a mosaic insert, which is meant to cover different clades. But we don't really know if by using and targeting some clades, it would be cross-reactive to others. And that's the type of things we are going really to learn when we do these clinical trials. But that's a very good point. It really depends on the antigen insert that we're putting in those vaccines, and they might be that they're more efficient by targeting different vaccine in different region, or perhaps we can have a universal HIV vaccine. That would be the best, but that's very hard to achieve.
Thank you. Any further questions? If not, then thank you very much.